I did a little research in my undergrad. We had a capstone course as during our senior year where we all flew down to Puerto Rico and got to design and implement a research experiment and then analyze the results and write it up in manuscript form. So I got to work with two other classmates, and we looked at a bioluminescent bay and how human activities were impacting the ecology of the bay. I met Michelle Baker, and it was just, she's seemed like an awesome mentor, and she was involved in this really exciting interdisciplinary project called iUtah, committed to water sustainability and across the state of Utah. So I thought it'd be a great opportunity to work with a lot of other individuals. I looked at how human activities are impacting stream biofilms. And biofilms are really just the slimy things on rocks that if you've ever slipped in a stream, it's probably one of those. And basically, they're just microbial communities that cover surfaces in streams. And they're incredibly important for the ecosystem. They do a lot of important things for nutrient cycling. And so I looked at how nutrient pollution and pharmaceutical pollution impact these microbial communities. I hope I can contribute to understanding what different types of bacteria are active for different ecological processes and the specific ones that are contributing or impacted by human activities. I love, we use this technique called DNA stable isotope probing, where we incubate microbial communities with an isotopically labeled substrate, so something that's just slightly heavier. And then you can, any bacteria that use that isotopic label will incorporate into their DNA and make their DNA slightly heavier. So you can use this really high-speed centrifugation to separate the DNA and identify who used that compound. So it's really incredible that we can detect, such a small change in the DNA. So the field days were pretty exciting. We were fortunate worked in urban streams, so we just were in Salt Lake City, say, but they could be long days. We'd want to capture, say, photosynthesis or different processes for these microbial communities. So we'd start out, there are some 4 a.m. days where we'd leave Logan with the whole car loaded up to the brim with coolers to collect samples. We'd get to the stream, put on our waders, and sometimes you go down a steep embankment, but once you're in the stream working, you would never know you're in the middle of a city like Salt Lake City. It's just this totally different perspective that you miss when you're at street level. And then, we're mucking around in the streams, having fun. There's always someone working with you. Part of my experiment, I had to anchor the devices down in the stream with bricks. So it involved hauling a lot of bricks. But I was fortunate to work with a lot of great people who all enthusiastically pitched in, and at the end of the day, sometimes we're getting back at sunset, but you've spent a great day in the field, so I feel fortunate. Yeah. Well, definitely attend USU. There are a lot of great people and opportunities to explore what you're interested in. And just enjoy the graduate experience. It's a huge, growing opportunity and a lot of great people. I am working with the Learning Strategies Center at Cornell University, where I teach supplemental courses for the large introductory biology classes. So helping students succeed, maybe learn study skills, if not everyone comes with the same level of preparation, making sure that everyone can succeed.